So without further ado, I will introduce our first speaker, who is Philip Moore. And so Philip Moore holds a master's degree in biology and a graduate certificate in geographic information systems. He's employed with USDA, where his chief responsibilities are conservation programs, environmental compliance, and GIS. He's been a volunteer with Mountain True, which was formerly the Hiwassee River Watershed Coalition, since 2005, and an Adaptation volunteer since 2017. So his presentation is pre-recorded, and then we will answer questions afterwards.
Hi folks, it's really awesome and an honor to be able to present here to Confluence. My name's Philip Moore, and I'm going to present a project I did at NC State in fall 2019, looking at the impacts of climate change on stream temperature in the southern Blue Ridge. And I was really fortunate to be able to use a lot of Adaptation data for that. So as everyone knows, water temperature is critical to mountain stream habitat, particularly for dissolved oxygen and for good aquatic habitat. So a lot of national and international organizations model air temperature projections under various climate change scenarios, but they don't look at water temperature very much. So my thought was, can I look at some of our Adaptation data in North Georgia and southwestern North Carolina and develop some kind of a relationship between air and water temperature that could potentially be used to look at climate change impacts on water surface temperatures going into the end of the 21st century. So the study site is the upper Hiwassee River watershed in North Carolina and Georgia. It's a high elevation area in the mountains, varying from 385 meters to 1,600 meters elevation. These are cold water streams. If trout and other cold water fisheries aren't going to survive here, they're really not going to do very well anywhere in the southeast. So it's really a good area to study to see how it's going to weather climate change. And we've got 100 sites here that have been monitored for a long time by volunteers with Adaptation. So I pulled that data from Adaptation, and of course, most folks are volunteers. They know we're looking at chemistry, temperature, and E. coli data. And so my main concern obviously was air and water temperature, so I was able to pull it for those sites. Now, as far as climate projections, that came from Oregon State, the PRISM climate group, 30-year model, 1981 to 2010, mean monthly air temperatures for March, April, May, June, July, August, September throughout the continental United States. So that was essentially where I got my baseline data. And then for projections, that came from the National Climate Assessment, mean monthly air temperatures as projected for March through September 2099. 1 GCM, that's global circulation model, and 2 different scenarios of a moderate carbon emission scenario and a high carbon emission scenario. So from the sampling data from Adaptation, I developed a regression model essentially to predict water temperature given current months air temperature and potentially looking at past months air temperature as well. And I used a method in GIS called map algebra to work on, apply that to raster data, to apply regression to historical and projected air temperature rasters, and therefore to predict the water temperatures. Then I also predicted how water temperature would change simply by taking the projections and subtracting the current data. And then I applied these water temperature changes to sampling points to get the predicted site temperatures under a moderate carbon and a high carbon emission scenario, and I joined this to the stream reaches. So the results essentially said that the current month's air temperature plus the prior month's air temperature resulted in a decent regression, so R square of 0.72. So essentially the predicted water temperature was based on this equation looking at the current month's air temperature and the previous month's air temperature. 2 months prior was really not statistically significant, so I omitted that one. So I'm going to just cycle through these results here for a couple times. There's normal, the moderate carbon, and the high carbon scenario. And so I kept the color scale the same throughout so that you can really compare the difference under these scenarios. So, and here again, applied at the stream reach level, under 19 degrees Celsius is green, that's supporting. 19 to 22 degrees Celsius is marginal, and above 22 degrees is unlikely to support cold water fisheries. So unfortunately for the hottest month, August, you see a lot of red under either scenario. Under the moderate emissions, it's about 1/3 marginal, 2/3 non-supporting. And under the high emission scenario, it's like 16% marginal and 84% non-supporting. So the results, sobering as they are, fit with other studies such as Selbig and Caldwell et al., which essentially suggested that unless we do some major mitigation in regards to climate change, we're going to lose a lot of our cold water fisheries in the mountains. So what we could do to improve the project that I did would be, one thing, replicate Caldwell and his colleagues by performing individual regression analyses for each site and omitting sites with fewer than 20 months of data. So actually apply regression site by site rather than overall throughout the region. And we'd hopefully get a better R square and reduce standard error. Also consider and control for solar irradiation and incorporate other global circulation models in addition to the one that I used. So finally, there's going to be a challenge with the future of climate change if we don't act pretty aggressively to mitigate adaptation, like planting buffers, removing dams that are, not really of any value, and removing ponds to kind of mitigate solar irradiation, mitigate higher temperature by reducing solar radiation. That alone is probably not going to be enough. We'll actually have to make a more of a national and global effort to mitigate. At the same time, though, this study could be valuable in order to prioritize adaptation work to, which streams are going to be the biggest bang for the buck, which ones are going to be worth the time and expense to actually preserve the aquatic value. So I appreciate your time, and I now look forward to answering questions.
Hello, everyone. Thank you, Philip, so much for such an interesting presentation. If you could join us now, that would be great.
Thank you.
I see, there you are. Awesome. Well, I haven't taken a look at the Q&A yet, but do we have any questions in there, Jackie?
All right, so for our attendees, if you have any questions, go ahead and you can enter them into your Q&A function at the bottom of your screen.
I had a few questions. So you used the air temperature to model what the water temperature would be in the future, right?
That's correct, right.
Okay. And I was wondering, is that kind of like a common method used, or just some people also incorporate other factors? I can imagine rainfall might be another factor that could influence water temperature and things like that?
Sure. I don't know actually how common that is. It was based on the data I could find and based on the projections, future projections. The best I could come up with I couldn't really find any projections related directly to air temperature. So I looked at this model of trying to tie air and water temperature together and see how well that worked. So, and of course it got, I got a decent regression, not a great regression. And I'm sure by incorporating those other factors, especially rainfall would be a big one that would improve the model. But so essentially for a semester project, that was what I came up with. But it is very hard, actually, in my experience of the research I did, to find projections that are directly related to water temperature.
Thank you. Any questions?
No, we don't have any questions in the Q&A. I was just curious.
We have a kitty with us today.
Oh, surprise special guest. Yes.
This is Tessa.
Oh, hello, Tessa. I was just wondering why. So you used the group with Oregon State Prism.
Right.
We used their climate data. I was just curious, why you chose them. I'm not familiar with them, and I didn't know if they do specialized work, or I was just kind of curious.
Right. That was the most comprehensive records I could find in terms of climate records, as far as in a raster format also. It was I was needing something that I could work with pretty quickly rather than develop GIS rasters. They really already had that data ready to go. So I could just download it and clip it to the watershed and go with it. So that was the most comprehensive data I could find when I started searching.
Gotcha. Cool.
I had another question. Do you have climate adaptation efforts already begun in regards to cold water fisheries and preparing for climate impacts in North Georgia?
Well, definitely through the Watershed Coalition, what used to be the Watershed Coalition, now Mountain True, we're looking at obviously adapting through better riparian buffers and better shading. That's critical. It's going to be keeping the shade on those streams, and so they're definitely looking into that. And then, but at the bigger scale of mitigating, that's something that is going to take a lot of folks working together. So that's more of an advocacy type position that we all get on the same page and kind of get serious about how we handle our emissions.
Yeah.
We do have a question here from Callie Moore. So she wants to know, are you able to provide a list of streams that would be the highest priorities for adaptation?
Okay. The highest priorities for adaptation, rather than for adaptation would probably be some of our streams in the valley regions, and ones that Mountain True is already working on a lot with Brasstown Creek, Valley River, and Cherokee County, Brasstown Creek, and Clay County. So we certainly can pull that data and get that together and see what would be. I'd have to go back and just kind of look at the models and pull that data out stream by stream. I haven't done that specifically, but absolutely we can, with what I developed in my paper, looking at those areas that at least don't exceed that marginal rating, at least can stay marginal even in the hottest month, even in August. Those would be the ones to focus on, sure. So we could definitely look at that.
Yeah, that would be great. I don't know if you, Callie, and Philip want to exchange information or anything like that, but you could in the chat if you wanted to be able to contact
Sure.
But that's neat. That's always what I really like to see is science informing management, right, and informing conservation efforts. So that's really neat. I do that. Well, are there any further questions? If anyone has any more questions, please type it into the Q&A. Oh, sorry.
No, it's fine. We do have another question from Becca Reiser. I hope I said your last name correct. But she wanted to know, how do you think the magnitude of effects from warming air temperatures will compare to other things, such as changing land use and shade along stream corridors?
Okay, that's a very good question. Based upon the data I saw and what I looked at so far in my model, I think both are very important. I mean, neither one can be ignored. So obviously changing land use, you know, even absent climate change is something that we have been dealing with, you know, as far as removal of shade on streams, more impervious surface, things of that nature, the number of ponds we have in the watershed, things like that, that really contribute to a lot of that thermal mass and a lot of that thermal heating without shade. So I think, you know, focusing on either one or the other does not work. I think we have to be very serious about both of those. So continuing to do the type of mitigation work we've done in the past, continuing to focus on shading streams, considering whether certain impoundments, and certainly some of them do, you know, some of them are generating clean energy and they have value that way, but considering maybe the ones that don't, we still need to keep those lakes, keep those ponds, you know, is there a way to mitigate that? So looking at that, and also at the same time continuing to both mitigate and adapt, essentially, for what we're dealing with.
Awesome. Well, thank you so much. I had Q&A open. I didn't see any further questions. But if you do have any that maybe you think of afterwards, you can also pop them in the Q&A as we kind of continue along. So thank you, Philip, so much. We're going to go ahead and move to our next presenter. And that is going to be Rachel. So again, this second presentation is going to be a live presentation from Rachel Pesaresi, I believe I'm pronouncing that correctly, hopefully, who is a recent graduate from the University of North Georgia. So today she'll be joining us to tell us about the change in macroinvertebrate abundance and diversity along the Etowah River. So welcome, Rachel.
Minor technical difficulty.
No worries, just let us know if we can help.
Can you guys see the PowerPoint okay?
We can, yeah. If you made it, yeah, if you make it full screen, we'll see it even better.
All right. Hello, everybody. My name is Rachel Pesaresi, and my research through my undergraduate is a comparison of the benthic invertebrates in the Etowah River. My PowerPoint is a direct copy paste from my poster because I wanted the most information if you had any questions based on what we have on there. It's not completed for the 2017-2018 portion, but essentially Jeremy Miller and Gabriel Pierce kind of brainstormed with Margie Flood, who is a professor at the University of North Georgia, and they needed some research hours, and she happened to know Dr. Tejan which through discussing with other people at Adaptation last year, some people know him, and he was an emeritus professor at the Georgia Southern State University, and he had his graduate research along the Etowah River after the nuclear aircraft labs were set up in different places along the Etowah. And he wanted to look at about 10 years after they had shut down and sold the property to the state of Georgia, what the water quality looked like based on what kind of invertebrates he saw. And we took over 50 years later. Jeremy and Gabriel went out and collected in the same spots, give or take 100 meters that Dr. Tejan did. And we went based on what we found. Another young lady and I took over and we separated out the invertebrates from the debris, and we started counting and later identifying what we found to see if in 50 years of the Etowah being primarily shifting from a place of possible nuclear waste debris to homelands, the growth in that area. And we wanted to know if it had been impacted negatively, positively, or if it hadn't, based on the diversity that we saw and the numbers. So what we found was a little disheartening. Between 1958 and 2017, we found that there was, in September alone, there was a 10 times more numbers for Dr. Tejan than what we found. And this continued throughout the 5 months that I was able to ID. This shows order, but I did take some down to family. And the other thing that helped us understand was that there is a loss of diversity in the families that Dr. Tejan found versus what we found. And the total numbers probably were more concerning than anything, because we took 3 separate samples at each of these 3 sites. So our numbers are 3 times the amount that he was collecting in how many times he collected. So this is not only 3 times the amount that he saw in 1958 and 1959, it's more of an exponential of 9 times as many. We saw some diversity lost, not as much as I was expecting when I did September's collection. Throughout the rest of it, it kind of got a little bit better, not better in a good way. And so it was mostly things that we would expect to see with bad water quality, based on what I understand and what we talked about. But the diversity that he saw was something of a healthy river system, whereas the diversity that we're seeing today, it's definitely questionable, which leads to the fact that we, as a research effort, decided that a really good route to go for the next generation of students is trying to figure out how the abiotic changed along the Etowah. But it does create a problem because this was 50 years ago. We might not have data for then. But I'm hoping that it does produce some good news throughout another research student taking over. And if you know anyone that's looking for research at the University of North Georgia, this is a really great involvement. It showed me a lot. But most of my research is if you guys have direct questions, because we did lose diversity, we did lose numbers. And yeah. thank you guys for listening.
Thank you so much, Rachel. That was, ooh, my screen is activating here. Okay, great. Yeah, so if audience members have any questions, please type them into the Q&A. I know I have a few questions, but I will maybe try to go first.
Let's see. Do we have any questions? Okay, so while we are waiting for people to enter in their questions, I think I missed this part. You probably had said it, but where did you get your data from 1958 and 59? Where was that sourced from?
Dr. Tejan himself knows my professor, so he came and he shared his numbers. He had them all written down in old books, so we at some point had to convert them to Excel. And he was actually the one that walked back with us and showed us the spots along the Etowah River, and helped us decide what would be a good place to take from based on what he did in 1958 and 59.
That's really cool. So why did he originally start collecting these numbers?
It was all about his graduate research. When he was going through grad school, he needed something that he found interesting and could get into, and invertebrates are definitely a way to go. They were a good way to tell, even if he didn't take samples of like the oxygen content based on what he did see in the numbers and what we saw comparing, you can kind of tell how the water quality has changed. So.
Yeah, thank you. I had two questions. The first is, you said that there were 10 times higher abundance in 58 than there was in 2017. And so I was wondering, for the diversity, what was like the multiplier of the diversity change between 1958 and 2017?
It's more of the numbers were 10 times. The diversity that we've seen, if you look at it only to family level, it's not as different. However, like he saw lots of dragonflies, the larvae. I could literally probably tell you confidently in the 5 months that I've sorted, I've seen 20. So that's where that number comes from, is that we were looking at his number and our number and comparing them specifically for September, which is kind of an odd month because there's not a huge emergence of new insects in that time period if they're sticking to that yearly schedule or bi-yearly. But the numbers were just not there on our end. We were seeing stuff that were 10 times less, or we were missing stuff from orders.
Mm-hmm. I realize I have two more questions. My first of the first series is that, so this is taking essentially 2 data points and not really looking at trends over time. So what if you had, for example, sampled like every year from 58 to 2017, what do you think that trend over time would be like? Do you think there would be kind of spikes, or would it just be a gradual decline based on your knowledge?
Based on what other papers that I had read in regards to long-term, yearly, or monthly trends, I think a steady decline is definitely possible. I think that some years were worse than others because we do have drought years that we see it across, Georgia or worldwide, that it does impact to this level. I think that in the temperature increasing steadily over the last 20 years, we have probably seen a little bit more of a decline. And I would gather to say, as far as, we started collecting in 2017 in September, I think if we went back this September, we would see a pretty dramatic increase for 3 years as well.
Mm-hmm. Yeah, which leads into my next question, which was, what do you think are the major factors that are contributing to this decline?
I think one of the major things is the growth in that area. Like I said, before the 1950s, it was farmland. So he only had about a decade to 2 decades of that nuclear aircraft laboratory being on along the Etowah. And he saw the numbers that he saw. If we had had data from 1940, I imagine that it would be twice as much as what it is today. I've looked into other little studies around this same area, and it's probably gonna increase in the number of decline just based on, even though we're being more cautious and more concerned with conservation, we've kind of started to destroy this population already.
Right. We have comments from Callie Moore, who says, a great follow-up would be to analyze land use using old aerial photos and current ones. The Farm Service Agency usually has good aerial photos from the 1950s. Google Earth also has some nice historical aerial images if that's something that you are interested in pursuing.
Yeah, I mean, I didn't think about going back to the 1950s, but that would definitely be something to look at, is how the land use has changed. And we discussed it a little bit, but this was a kind of a monster to take on.
Yeah, I can imagine. There's lots of different ways you can build on this question.
For sure.
Well, awesome. Well, thank you so much, Rachel. If anyone else has any more questions while we kind of continue, you can continue to put them in the Q&A box. And we will move on to our next speaker. So our third presenter is Kimiya, who is a senior at Woodward Academy in Atlanta. She has a pre-recorded presentation for us today that is about arsenic contamination in Atlanta groundwater. So Jackie will be pulling that presentation up momentarily.
Hello, my name is Kimiya Rezaiamiri. I am a high school student at Woodward Academy. I conducted an independent research project on arsenic levels in Atlanta area waterways. We have two types of arsenic, organic and inorganic. Inorganic arsenic is a toxic form and can cause health issues such as carcinoma, respiratory diseases, and skin diseases. The major source of inorganic arsenic is in groundwater. I collected and tested 3 samples from 9 rivers and streams within a 15-mile radius of the city. I used a quick rapid arsenic test kit, which had 3 reagents of zinc, tartaric acid, and nickel salts. These reagents will convert the arsenic to arsine gas. Arsine converts the mercuric bromide on the test strip to mercury halogens that appear with a color of yellow. The color changes from white to yellow, and the darker shade of yellow means higher levels of arsenic. 8 out of 9 river studies were found to be free of arsenic at 0 pph. However, 1 sample had slightly positive arsenic at 5 pph, which is still under EPA recommendation level of 10. In conclusion, I didn't see any dangerous level of arsenic in Atlanta groundwater. Thank you.
Thank you, Kimiya. She is on the call with us right now. I don't know if you want to put the video on, but you can if you want.
Hi.
Hi, awesome. Thanks for that presentation. If anyone, again, has any questions about the arsenic study that she did. Oh, we do have one. Okay. So Callie Moore is asking, how does the arsenic get into the groundwater?
Like by rocks and like when it waters, like from the rain. And then it goes into like streams and underground waterways.
Yeah, thank you. I have a question. What kind of inspired you to look specifically at arsenic contamination levels in groundwater? Like what were you thinking when you got involved at the beginning stages of this?
Well, I came across an article about how arsenic is like it can like do like damages to like humans and like living organisms. That's when I did the research.
Another question I have is, do you think that even though there's not arsenic in the groundwater, there could be arsenic in the surface water?
Probably. I mean, I did a test for that, but yeah, I got it. And maybe it's like another thing that someone could test for.
Kelly is asking, how did you obtain the samples?
So I visited each of the 9 rivers, and I took a bucket, and then I put the water in it 3 times. I took 3 samples from each river.
And what? So it was 8 of the 9 samples had 0 ppH, but that one had 5. And so where was that one river?
It was the South River.
It was the South River?
Yeah.
Okay. That's good to know. Jack, do you have any questions?
Yes, so do you have any plans to continue your research, or was this like just for an environmental science class?
I plan to continue it because I did it over 1 season, but I want to do it over like a year round to see if the season's effective.
Yeah, that would be cool to see, or I would want to know if it, changes depending on seasons, but.
Yeah, that's true. So when did you take these samples? What month was it?
November.
November. Nice.
Okay.
Yeah. Well, I don't see any other questions kind of coming in right now. If they do, then we can address them then. My Zoom is being a little finicky right now, but thank you, Kimiya. Yes, if anyone has any more questions, we'll ask you to come back on, but for now, it seems like everyone's questions have been answered.
Okay.
Our final and last presenter is Kelly Jackson, and this is also going to be a live presentation, and I believe her advisor is also on the call with us, Dr. Dyer. And so Kelly Jackson is a recent graduate from Clayton State University. During her chemistry studies, she became involved in the university's water research and Georgia Adaptation. And today she's going to be sharing her results from a chemical investigation into the contaminated waters of the headwaters of the South River, which is right on point because we just learned that the South River was the one that had arsenic in it. So it connects. Very interesting. So Kelly, if you want to turn your video and audio on and share your screen, that would be great.
Hello, my name is Kelly Jackson, and today I'm going to present my research on a small stream with big problems. So let's dive in and see why this water is so blue. First, I want to give a special acknowledgement to UCARE, Clayton State University, my Georgia Adaptation team, especially Dr. Kadani, Dr. Aubrey Dyer, and Hannah Palmer. Today I'll be discussing with you my research conducted on the South River and its headwaters. The South River begins in East Point, Georgia, and runs east through Atlanta, and it turns south and goes to Lake Jackson, and then joins the Ocmulgee River and empties into the Atlantic Ocean. Why is there concern? Well, the color may look pretty, but I believe we can recognize that this is concerning. It's in an urban area where families live and enjoy the outdoors. Parts of this river are guarded by fences, you can see here, but other parts you can just walk right up to, these ones here. The watershed map for the headwaters of the South River. The blue area shows the points that the water flows towards this green dot here, and that's where the start of the South River is officially. The sites tested are labeled here as 1, it's noted as the Tift right, 2 is Tift, 3 is Norman B, and 4 is Park Lane. And all of the feeder streams to the west are underground and not accessible for sampling. In exploring the watershed map history, a Class 1 hazardous site, which is the highest classification, and a Class 3 hazardous site both sit inside this watershed. The methods we used where the water was visible, we targeted on taking standard adopted stream test samples like pH, dissolved oxygen, temperature, conductivity, and biological measurements. This is the chemical kit we use, then the conductivity meter. You can see we did titrations, and this is how we tested for pH. These are the collected samples from the Park Lane location. They're both water and soil samples, and were collected in plastic bottles to avoid leaching from the glass containers, meaning that the acidic condition of the water may draw metals from the glass into the solution. Soil samples were sorted, ground with mortar and pestle, and digested with acid. Water samples were centrifuged as needed, and the soil results will not be discussed today. Not enough time. So here's a picture of some of the pH samples that we did. As noted, the water of the South River is very acidic. We found no life at any of our testing sites. The results on the site pHs were around 3.5 to 5.5. Shown here is 4.75 at Park Lane, 4.35 at Norman Berry, and 3.5 at Tift. The pH varied during our courses of testing, but it was never higher than 6. pH meter was used in the lab to notate the pH samples sent for analytical measurement. We took the samples to the University of Georgia and ran them through an ICP OES. The concentrations were compared to values calculated from the EPA approved water quality standards. Each of the bar graphs contain 2 horizontal lines. These include the chronic and acute calculated levels. Some graphs do not show the horizontal line due to the high concentrations of that element in comparison. A water hardness of 100 was used in this calculation. In general, we observed a decrease in concentration at the Park Lane sites, which was about 1 mile downstream from the other sample locations. With the cadmium, you see the first 3 are all above the acute line. The Park Lane was below the level that you can measure. All right, the zinc levels were all above acute. The copper levels were above acute on all 4 locations. And the first 3 samples are higher than chronic levels of lead, and the first 2 samples higher than acute levels of lead. And the Park Lake sample was below detection. There's no set standards for ions in surface water, yet this is worth noting the high concentrations of fluoride in the Tift site. The concentrations appear to drop off by the time they reach Park Lane. But this chart shows the numerical comparison of the results with the pHs listed. And you can see here that the detection limit for cadmium is still above the acute line, and the detection level for lead is above the chronic line. So it's possible that both of these can still fall in a very hazardous concentration. These are the 2 outflow pipes that flow into the Norman Berry site. We didn't find the one magic thing that makes the water appear milky blue. If we did, we didn't understand it. We have several ideas and directions to go to continue the research. In fact, Clayton State is establishing a water testing lab to further the research. The water chemistry coming out of each one of these is very different, both too toxic to support life, but they joined together to start the South River. So this picture was taken at the Park Lane site. You see here it is fenced off, but it is next to a park and across the street from an elementary school. And so this is one of the places that it was fenced off, but the other places we easily could just walk up to be able to sample. Questions? And there's my calculations and references.
Thank you so much, Kelly. That is very scary, actually, those results. For sure. We have one question already in the Q&A, and if anyone has any others, please put them in there. So Callie also thinks that these are kind of scary results. She asked, are these sites hazardous waste landfills or industrial sites or what? Because
historically, over 100 years of chemical plants, there is a landfill and possibly a burned down cotton mill and manufacturing there.
That was going to be my question too. So particularly the Tift site looked like it had highest levels of almost everything.
Mm-hmm.
So is that where one of the class 1 hazardous?
The class 1 hazardous site is the Tift site.
Okay, yeah. It is.
And there's a landfill there now.
Well, that's very scary. And I have two more questions. I know you said you weren't gonna go in super detail about the soil results, but could you provide like a quick summary? Were they approximately kind of the same or similar to the results of the water testing?
I did not prepare for that, but they were both, there was some really high stuff, but there's not really a baseline to know is that high good or bad. So we just didn't include that in the results.
Right. Mm-hmm. Because you said that there are no standards for ions, and those ions were cadmium.
No, the ions were fluoride, chloride, sulfate, and phosphate.
Okay, gotcha. Mm-hmm. We have another question from Rachel, yes, who is asking, any thoughts on why there's so much CU in Norman B?
Well, the pH there is quite low where they both meet. We have some theories about what's coming from the side that we weren't able to test from, but I think it's where they join and they mix. There's just a special chemistry that happens there. So copper, aluminum, sulfate.
Yeah. If you were to be providing sort of recommendations for cleanup and mitigation, what would you say?
I'm not sure. Yeah, I don't know.
Awesome. Well, does anyone else have any questions? If you do, please type them into the Q&A box. We do have just a few more minutes before we're going to—
I guess like a wetlands area is what they had discussed, but the airport had shut that down because of birds.
Oh, okay. Interesting. And then what are the ideas and directions that you see kind of this project going towards? You had mentioned something.
I'd like to try to recreate it in the lab to see if we can get this type of conditions where it is like this glowing white blue in the water, trying to mimic those conditions. And then we need to dial in on those two that were below detection and maybe go ICP MS to try to get those more dialed in and see what the actual concentrations are.
Awesome. Well, best of luck with all of that. I hope that you're able to do all that and that goes well, so you can determine what specifically is making this water kind of that milky blue color, so it can fix it, because it's very sad that these elementary school students cannot get in the creek.
Definitely don't get in the creek.
Yeah. All right. Well, if anyone has any more questions, oh, we do have one. Okay. Callie Moore says, I think that EPA Superfund program would need to answer Jen's last question. I know. I was just thinking kind of broader answers, but Superfund should definitely get on that. Are there any Superfund sites within that area?
Dr. Dyer, what was the name of the park company? Do you remember?
Oh, I don't remember. I think there's a Superfund one where it was like further north, and it's, I think, where they used to do the treatments for power poles. I can't remember what it was called, but my mind's gone blank, where they put the tar and things on those power poles. I think that's a Superfund site, and that's not far from there. So it's very sad, all of this contamination in that same area where people live and schools are, and yeah, it's very sad. And then where this river starts, and the Flint River is just on the other side of the railroad tracks. So a lot of interest.
The company that did the park and like had to go get it tested and approved, they sent their soil samples to UGA, but they were testing for like the health of the soil as far as like growing things. They didn't test for any heavy metals or anything.
Okay, gotcha. Well, it sounds like that they need a lot of remediation efforts there, and hopefully your research can help inform kind of where to best focus and what's causing the issues. So thank you so much for sharing that with us. It seems that we've lost Jackie, which she told us that there was a big storm coming in, so that's all right. We will just kind of conclude without her. So yeah, that is all of our presenters for today. So if everyone was in the same room, we would be giving a big round of applause. So this is me just giving a round of applause to you. Jackie would be joining me, but she's not here. Oh, she's back. That's great. But we just have a few closing remarks and reminders. The first is that we have our Adaptastream social in just about an hour. So it's almost 6 o'clock. We have a social tonight that starts at 7 and it goes until 8:30. So if you are interested, you should totally join us. The last one was really fun, so much fun that a second one was requested. And again, starts in an hour. So if you're interested in that, maybe just go grab some food, grab a beverage of your choice, and join us back in virtual land at 7. There will be links for that put in the chat. And then if you haven't already, please join our Confluence Facebook group. We're coming up on the last week of Confluence next week, which is crazy how quickly this month has flown by, but we're still trying to provide people a kind of a virtual space for networking and talking with each other and providing updates on what everything that's going on. So that is a place where we do all of that. And there are lots of great more sessions that are coming up this next week related to some of the soil contamination work that we were talking about today. There is going to be a session about soil contamination around Atlanta metro area by Dr. Ari Sakawa from Emory University on Wednesday. I believe it's a noon session. So if you want to hear more about that, be sure to tune into that session. But if for some reason Wednesday at noon is not good for you, then you can watch that session afterwards because we record them all and post them on our website. Jackie, are you with us?
Yeah, I'm so sorry. I am on my phone's hotspot now. So I apologize for that. We had some thunderstorms roll through and I think lightning may have caused me to black out. But Jenna, have you entered the chat yet for the links or?
I have not. I can do that right now.
Oh, okay.
Now they're in there. So if you're interested in that soil session I was talking about, you can that session registration page link and that will take you to where you can find it to register for it. Again, it's on Wednesday. And last but not least, we have a silent auction as per Confluence tradition. And so this year, since we can't be in person, the silent auction is going to be online and you can bid until August 31st. There are some really awesome items, including winery tours and stays in mountain cabins and tickets to theater productions within Atlanta. So there's lots of really diverse things that you can find on there. And then lastly, when you leave you're going to be directed to a survey or a page that will ask you to participate in a survey. And so it's just a quick 5-question post-session survey. And we would love for you to take the time to fill that out and provide us feedback as we continue to improve and hone in on what works best in this virtual Confluence space. So thank you again, everyone, for joining us on this Friday evening. And we hope to see you at another Confluence session again soon. Hopefully, the virtual social in an hour, but if not then maybe next week, and have a great night. Bye everyone.